Hello, everyone. We are now down to our last chapter discussion for the biochemistry. We are now in the chapter 6 for the biochemical energy production. So, in the next session, we will be discussing the following item from metabolism up to most likely on oxidative phosphorylation. We will just see if kaya sitong dime and for the final exam coverage. So let's now discuss what is a metabolism. Metabolism is the sum total of all chemical reactions in a living organism. And metabolism is also the source of energy for the functioning of the human body. It's also needed for many of cellular processes, such as protein synthesis. Second one is DNA replication, RNA transcription, and membrane transport. And these processes need energy because the cell must use it to build molecules and to copy genetic materials and move substances across membranes, and all of which are essential for growth, repair, and normal body functions. And there are subtypes of metabolic reactions. One is catabolism, and the second one is anabolism. Catabolism is when all metabolic reactions in which large biochemical molecules are broken down into smaller ones. It is usually the energy is released in these reactions. For example, is on the oxidation of glucose. Because in this example, in the oxidation of glucose, the molecule is broken down into smaller molecules. That is the carbon dioxide and water, releasing energy in the form of ATP or adenosine triphosphate that cells use for various functions. By the way, you can still take down notes if there are important key points that I will be sharing with you that is not present in the presentation. And the second metabolic reaction is known as the anabolism. Once we say anabolism, it's all metabolic reactions in which smaller biochemical molecules are joined to form larger ones. For example, we have here synthesis of proteins. So, what usually happens in the synthesis of proteins? In this manner, small molecules called amino acids are joined together to form larger protein molecules, and a process that requires energy to build and maintain cell structures and functions. That's how synthesis of protein work. Another one, let me show you the figure 23.1 on the processes of catabolism and anabolism. So in this figure, um, this actually shows the relationship between catabolism and anabolism. And in this figure, the two main parts of metabolism is actually categorized. So, one is uh, catabolism, and the second one is anabolism. In the case of catabolism, in this figure, the large molecule are broken down into smaller ones. Okay. Uh, and what happens here is that there is now a release of energy so that the cell can actually use this. However, in anabolism, it uses up the release energy to build larger molecules from the smaller molecules, therefore supporting its growth, repair, and maintenance of the cell. And together, these processes, both the catabolism and anabolism, maintains the balance of energy and materials within living organism. So what are the metabolic pathway in the metabolism? This pathway is known as a series of consecutive metabolic reactions that is used to convert starting material into an end product. And there are two types of metabolic pathways. First one is known as linear. Again, this is known as linear. And the second one is known as cyclic. And its metabolic pathway has its major um, point of entry, in which all forms of life are similar. For example, in this figure actually shows the two types of metabolic pathways. One is, again, the linear, and the second one is the cyclic um, pathway. Okay, so in the linear pathway or linear metabolic pathway, reactions proceed step by step in one direction, okay? From this point up to this point, from enzyme 1 up to enzyme 3, where each product becomes the substrate for the next enzymes, okay? Highly dependent from previous enzymes until a final product is formed. In this case, our final product is product delta or D. Okay. And in the cyclic metabolic pathway, the final product is used to regenerate the initial molecule, allowing the cycle to repeat continuously. And for example, of the cyclic metabolic pathway is we have the Krebs cycle in a cellular respiration. So let's have a practice exercise. Kindly classify each of the following chemical process as anabolic or catabolic. Right, so what's our answer? Okay, so for the synthesis of protein from amino acid is known as anabolic. Formation of triacylglycerol from glycerol and fatty acid is known as anabolic. Hydrolysis of polysaccharide to monosaccharides is catabolic. And formation of nucleic acid from nucleotide is known as anabolic. And what are the two types of metabolic reactions? Okay, it's the anabolism and catabolism. So let's now proceed to metabolism and cell structure. Okay. So metabolism and cell structure. I think you already have a knowledge on this in your senior high school or in your previous years. If you were first year from the other section for MLS 2I. Um, it's the knowledge of the cell structure that is essential for understanding metabolism. And in the case of prokaryotic cell, no nucleus and found only in bacteria. And prokaryotic cell uh, is presence. There is a presence of a single circular DNA molecule near the center of the cell called nucleoid. Therefore, um, in this prokaryotic cell, we can actually say that the DNA is present in a singular molecule because they lack a nucleus to contain and organize multiple chromosomes. And this circular DNA efficiently carries all the genetic information needed for the cell. Uh, to function and to easily replicate within the cytoplasm. But how about the uh, eukaryotic cell? In the eukaryotic cell, we can say that the DNA is enclosed in a nucleus to protect it from damage and to better control gene expression and cellular activities. And the nuclear membrane separates the genetic material from the rest of the cell, allowing more complex regulation and organization of cellular processes. So we have here our figure 23.3, the schematic diagram representation of eukaryotic cell. In this figure, it is actually highlighting its major organelles and their functions. The nucleus, we say, contains the cell's DNA and it controls its activities. While your cytosol is the fluid where many metabolic reactions occur. And how about your ribosomes or your small dots? These are actually responsible for protein synthesis, while your mitochondria actually produce energy for the cell to survive. And your lysosomes is one. Your lysosomes break down waste material, and the plasma membrane surrounds the cell, controlling the movement of substances in and out of the cell. So we're now on eukaryotic cell organelles and their function. So your plasma membrane is also part in the cellular boundary. And as I've mentioned a while ago, your cytoplasm is a water-based material of a eukaryotic cell. Uh, yeah, your cytoplasm is a water-based material of your eukaryotic cell, and your mitochondrion generates most of the energy needed for the cell. I think I forgot to mention on the lysosome part that it actually contains hydrolytic enzymes needed for cell rebuilding, repair, and degradation. And yeah, else. Um, your mitochondrion. Uh, Let's let's explore your mitochondrion. Although I have shared it a while ago, on a brief statement or brief description, but let me discuss this clearly. That mitochondrion is an organelle that is also responsible for the generation of most of the energy for a cell. And in the outer membrane, there is known as permeable to small molecule. Therefore, um, 50% lipid and 50% proteins. And the inner membrane, it's highly permeable in the mitochondrial part. And it's 20% lipid and 80% protein. And it's actually folded to increase the surface area. And the synthesis cores the ATP. So yeah, so we say that synthesis of your ATP occurs in the inner membrane of the mitochondrion because the enzymes and proteins needed for the electron transport chain and the adenosine triphosphate synthase. It's folded structure called as cristae, and it increases the surface area, allowing more space for this reaction to produce large amount of energy efficiently. So another exercise, what cellular organelle is responsible for the generation of most of the energy for the cell? So what's your answer? Okay, that's mitochondria. All right, let's now continue. We're now down to the important nucleotide containing compounds in metabolic pathways. So we have here your adenosine phosphates. Adenosine phosphates is classified among the following items. So we have adenosine phosphates, adenosine monophosphate or AMP, adenosine triphosphate or ATP, and adenosine diphosphate or ADP. So your AMP is actually a one phosphate group that's a structural component of your RNA. And your ADP is the key component of metabolic pathway that's it's composed of two phosphate group. No? And your ATP is a three phosphate groups. That's the key component for still the key component for metabolic pathways. So let me now show you the reactions. So we have here a phosphoryl group that is derived from a phosphate ion. So when it becomes part of another molecule. So in here we have here your reactant and your product. So what happens in this reaction for in your reaction number one? So we have here your ATP plus H2O that results to your ADP rather, your um, phosphorus, your hydrogen ion, and the energy. So this is now the hydrolysis of your adenosine triphosphate um, to adenosine diphosphate. That's an inorganic. Your P sub I P sub I is known as your inorganic phosphate. So what happens in this reaction is it actually releases energy that's approximately 30.5 kilojoules per mole or approximately 7.3 kilocalories per mole under the standard conditions. So what is the significance of this reaction? So this is the most common reaction in cells for energy transfer. And the released energy powers the cellular to work, such as your muscle contractions, active transport, and biosynthesis. One example for this reaction is your uh, exer, exergonic reaction or energy releasing. So, pwede na to ninyo isulat ang ako ang isulat, no? I mean, istorya nga wala sa presentation. So, another reaction is we have your ADP. This is now the hydrolysis, the reaction of the hydrolysis of your adenosine diphosphate going to your AMP or adenosine monophosphate and including your inorganic phosphate. So what happens here is it also releases energy, slightly less energy than your ATP and your ADP hydrolysis. So what is the significance of this second reaction? These reactions can occur when energy demand is very high. It is less common, but important in regulating energy balance, especially that that these processes consume large amount of your adenosine triphosphate. So we have the third reaction, right? We have your ATP uh, um, moving to your AMP. Okay, so this represents the complete hydrolysis, kining third reaction, complete hydrolysis of your ATP to AMP and your two inorganic phosphate. It actually combines the two-step above, no? From your ATP moving to your ADP plus the inorganic phosphate and your ADP plus your AMP and inorganic phosphate. So, what is the significance of this reaction? In this manner, it produces the greatest energy released per molecule of your ATP. And seen in this process requires a large rapid energy input such as amino acid activation during the protein synthesis or signal transduction pathways. Example is your CAMP formation. All right. So another interesting reaction. So we have here your reaction, no, in the cellular uh, manner. We have here your ATP functions as both a source of a phosphate group and a source of energy. And we have here example, the conversion of your glucose to glucose 6 phosphate. Okay? Sige. So let me discuss to you the component of your glucose. So your glucose is actually a 6 carbon sugar that serves as a primary energy source for cells. And your ATP here, or your adenosine triphosphate, actually donates a phosphate group of PO3, PO subscript of 3, and then exponent of 2 negative, and that actually provides energy for the reaction. And in this, um, your reaction, in your hexokinase. This is the enzymes that catalyzes the transfer of the phosphate group from ATP to glucose. We also have G6P or glucose 6 phosphate. This is the phosphorylated product of your uh, glucose. And yeah, I forgot to mention your ADP. ADP is formed when your ATP loses one phosphate group. No? So minus one. So from tri, it will now turn to be di, triphosphate to diphosphate. So what happens chemically, in this reaction? Your hydroxyl group, that is your OH, that is colored in blue, on the 6 carbon of glucose is replaced with phosphate group, or your O dash PO3 to negative. And your phosphate group is now transferred from ATP to glucose by the enzymes, no? That is known as hexokinase. And the reaction consumes one molecule of ATP, which is converted to ADP, right? So, what is the role of other nucleotide triphosphate in metabolism? One is for the UTP, or uridine triphosphate, involved in carbohydrate metabolism. And your GTP, or guanosine triphosphate, is involved in protein and carbohydrate metabolism. And your CTP, or cytidine triphosphate, is involved in lipid metabolism. We also have your FAD or FAD, the flavin adenine dinucleotide. This is a coenzyme required in numerous metabolic redox reaction. It is also a flavin subunit that is active form which gains hydrogen atoms when FAD is converted to FADH2. Ribitol is a reduced form of your sugar ribose. Okay, so let's now proceed to your structure of your FAD and your NAD plus. So let's now start with your flavin adenine dinucleotide. Your FAD is made up of three main parts. One is flavin, the second one is ribitol, and the third one is your adenosine diphosphate. Your flavin is derived from your vitamin B2, okay? Or it is also known as your riboflavin. It is the site that accepts or donates hydrogen atoms. Whereas your ribitol is a sugar alcohol that links the flavin ring to the phosphate group, no? Moving to your um, ADP. While your ADP contains now your adenine, ribose, and two phosphate group. Okay. So in short, your riboflavin plus your ADP, again, your riboflavin plus your ADP will now result to flavin adenine dinucleotide or FAD. So what is the primary function of your flavin adenine dinucleotide? It actually acts as a coenzyme for oxidative enzymes, that is your flavoproteins, in reactions involving hydrogen atom transfer. It alternates between FAD. FAD is in your oxidized form, while your FADH2 is the reduced form, accepting two hydrogen and two electrons. And the overall reaction in this structure is we have here your FAD plus your 2H plus two electrons will now result to your uh, FADH2. Okay. So what is the role of your um, FADH2 in metabolism? Your FADH2 um, participates in the citric acid cycle or the Krebs cycle, such as the conversion of succinate to fumarate by the succinate dehydrogenase. So your FADH2 will now then later donates its electrons to the electron transport chain, helping produce adenosine triphosphate. So let's now proceed to the second reaction. We have here your nicotinamide adenine dinucleotide or NAD+. Your NAD plus is actually contained of one is nicotinamide, we have the ribose, and we have your adenine. Your adenine is a nitrogenous base, and your ribose is a sugar that is attached to nicotinamide. Nicotinamide. And your nicotinamide is derived from your, this is actually derived from niacin. That's vitamin B3. This is the reactive site where the reduction occurs. So what is now the function of your NAD plus or NAD plus? This is it actually acts as an electron uh, carrier alternating between the NAD plus and your NADH or NADH. So what is the role of your NAD plus in metabolism? The function in the function of this NAD plus is it actually helps the catabolic reaction, such as glycolysis, the citric acid cycle, and then the oxidative phosphorylation. Your NADH carries electrons to the electron transport chain that will now generate to your adenosine triphosphate. All right. So I think I have mentioned some of the item, no? Bullet one, two, and three. So let me just emphasize that your FAD is a typical cellular reaction in which FAD serves as oxidizing agent, involves conversion of an alkane to an alkene. And your NAD. Um, this is a typical cellular reaction in which your NAD plus serves as an oxidizing agent, is the reoxidation of the secondary alcohol to give a ketone. All right, so we now have a subunit structures of your NAD. So the three subunit for your NAD, we have nicotinamide, ribose, and ADP. And six subunit structure for is we have nicotinamide to ribose to phosphate, and from phosphate to ribose to adenine. So how about your coenzyme A? This is a derivative of vitamin B pantothenic acid. And this is an active form of coenzyme A, is the sulfhydryl group, which is it has an SH group in the ethane ethiol subunit of the coenzyme. And your acetyl coenzyme A form when acetyl group bonds to coenzyme A, SH group, via a thioester bond. So we have here your three subunit structure for coenzyme A from nicotinamide, ribose to ADP, and for the six subunit structure from nicotinamide, ribose, phosphate, back to ribose again, and then moving to adenine. So there are now classifications of metabolic intermediate group. So your metabolic intermediate compounds can be classified into three groups based on their functions. Okay, for first intermediate group is intermediates for the storage of energy and transfer of phosphate groups. So we have ATP, ADP, and AMP. And the intermediates for the transfer of electrons in metabolic redox reactions is we have um, FAD to FADH2 and from your NAD plus to NADH. And the intermediates for the transfer of acetyl groups is your HS coenzyme A to acetyl S coenzyme A. So I think we are now ready for some exercise. So what intermediate molecule in metabolic reactions is responsible for producing energy in human body? So what's your answer? Okay, and that's ATP. The second one is, why is ATP the best energy source for human beings? Right? What's your answer? All right. It has an intermediate value in free energy and it undergoes slow hydrolysis in an aqueous environment. We also have um, important carboxylate ions in metabolic pathways. All right. Carboxylate ions are your metabolic acids. This is a polyfunctional acids formed as intermediates of metabolic reactions. And there are five uh, such acids that serve as substrates for enzymes in metabolic reactions. Okay, we have here the three succinic acid, the derivatives of C4 diacids. We have here fumarate, oxaloacetate, and malate. And we have also two glutaric acid derivatives, that is C5 diacid. We have alpha ketoglutarate and citrate. So we have here the structural formulas for polyfunctional carboxylate ions. So please take time to check muna the succinic acid and your glutaric acid. All right. So let's now start with your succinic acid. Your succinic acid is a four carbon diacid, and glutaric acid. We also have five. Your glutaric acid is a five carbon diacid. That's actually these both acids were formed from various biological derivatives from such as hydroxy, keto, hydroxy derivatives, keto derivatives, and unsaturated derivatives for succinic acids or compounds that are also intermediates of the citric acid cycles. And these derivatives, such as your malate, your fumarate, your uh, oxaloacetic acid, acetic acid, what else? Your citrate, um, okay, this one, your citrate, and your alpha um, ketoglutarate play key roles in cellular energy production and metabolisms. So in the metabolic pathways that is shown on this figure um, is actually crucial because they represent key steps in the citric acid cycle or your Krebs cycle. That is your central pathways of energy metabolism. And these reactions convert nutrients into ATP, your ADP, ATP, NADH, and FADH2, which provides energy for cellular processes and also produce intermediates that is used for synthesizing amino acids, fatty acids, and other essential biomolecules. So let's now have an exercise. So which of the following structural descriptions for metabolic carboxylate ions that are derivatives of succinic acid is incorrect? So please take your time to look for answer. All right, what's your answer? Okay, that's fumarate, contains a carbon-carbon double band in a cis configuration. So let's now proceed to high energy phosphate compounds. Several phosphate containing compounds found in metabolic pathways are known as high energy compounds. And these high energy compounds have a greater free energy of hydrolysis than a typical compound. And these compounds contain at least one reactive band that is called as the strain band. And it is known as the energy. The energy to break these bonds is less than a normal bond. And there are more negative, the free energy of hydrolysis, and the greater the band strain in terms of the energy bounds. Typically, the free energy release is greater than 6 kilocalories per mole. That's the indicative of band strain. And the strain bands are represented by the sign squiggle or approximately. CK. Let's now proceed to um, table 23.1, or the different free energies of hydrolysis of common phosphate containing metabolic compounds. The first type is we have the enol phosphates. Example for enol phosphate is phospho enol pyruvate. Okay? Its average free energy of hydrolysis in terms of kilocalorie per mole is approximately um, 14 kilocalorie per mole. This means that these have a very high energy phosphate bonds due to the unstable enol 4. And the hydrolysis produces a stable ketone that form from your pyruvate, which derives the large energy release. Okay? Sige. And your phosphoenol pyruvate is one of the highest energy compounds in metabolism and is very important. And it's very important in the ATP synthesis during glycolysis. The second free energies of hydrolysis is known as acyl phosphates. Acyl phosphate, one example is we have the 1,3-biphosphoglycerate acetyl phosphate. Its average free energy is approximately negative 11.3 to negative 11.8 kilocalories per mole. These acyl phosphates contains a high energy acyl phosphate bond that is your um, CO dash CO dash your phosphate. Okay, the hydrolysis releases energy because it forms both catabolic or carboxylic acid and inorganic phosphate that is both stabilized by resonance. And your acyl phosphate has important intermediates in glycolysis and other metabolic pathways where they transfer phosphate groups to ADP to form your adenosine triphosphate. Another one is your guanidine phosphate. Example for this is your creatinine phosphate and your arginine phosphate. Approximately, it has a negative 9.9 to 10.3 kilocalories per mole. And its primary function is to store energy in muscle and nerve cells for rapid adenosine triphosphate regeneration. And the nitrogen containing the nitrogen phosphorus bond now releases energy reservoir, replenishing your adenosine triphosphate during the short bursts of intense activity. So we have the fourth one. All right, please read silently the fourth, fifth, and the sixth one. Um, yeah, let's just have a water break. So you may just scan this and read some of its example and details. All right. Um, can you still hear me? Sige, where are we? I think we are now on the fourth example, that is your triphosphates. Example here is we have your reaction from your ATP to AMP uh, plus your inorganic phosphate. So approximately we have here negative 7.5 to 7.7 kilocalories mole. And the primary function of your triphosphates is it actually contains two high energy phosphoanhydride bonds. And the hydrolysis relieves your electrostatic repulsion among negatively charged phosphate groups and yields stabilized products. And in this manner, your ATP serves as your universal energy currency of the cell, driving many biological reactions. So, how about the fifth one? So, we have here your diphosphates. Your diphosphates is we have here a set of examples. We have here your ADP um, that undergoes reaction of that's been moved or converted to AFP to inorganic phosphate, and your PPI to 2 inorganic phosphate. So what happens here is it actually contains one phosphoanhydride bond, and the hydrolysis of pyrophosphate or your PPI is known as pyrophosphate, is often coupled with biosynthetic reactions to make them essentially irreversible. And your ADP hydrolysis also contributes to the cellular energy transfer. And the sixth example is your sugar phosphates. Example for this is we have the glucose 1 phosphate, fructose 6 phosphate. What else? Your AMP to adenosine plus inorganic phosphate, or glucose 6 phosphate, and glycerol 3 phosphate. So in this manner, in this reaction, it has phosphoester bonds, no? Let's say phosphoester bonds, which are low energy compared to other phosphate compounds. So in this case, hydrolysis releases less energy because the reactants and products are similarly stabilized. And it also serves as intermediates in metabolism, example, glycolysis, gluconeogenesis, and for energy trapping inside cells, rather than as a direct energy sources. All right, another short activity. So how many strain bonds are present in an ATP molecule? So please answer it. You may get your notebook with you. So your answer is 2. So let's now proceed to the overview of the biochemical energy production. Energy needed to run the human body is obtained from food via a multi-step processes involving several different catabolic pathways. And there are 4 general stages in the biochemical energy production processes. One is digestion. Second one is acetyl group formation. Third is citric acid cycle. And the fourth one is electron transport chain and oxidative phosphorylation. However, each stage also involves numerous reactions. Stage 1 for digestion. It begins in the mouth, no? And the saliva that contains starch digesting enzymes continues in the stomach that has a gastric juices and is completed in a small intestines. It now then resulted in a small molecule that can cross intestinal membrane into the bloodstreams. And the end products which are now absorbed and transported to blood cells, it is in the form of glucose and monosaccharides from your carbohydrates. We now also have your amino acids coming from your proteins and your fatty acids and glycerols from fats and your oils. Those are your end products. So for step 2 or stage 2, your acetyl group formation. So in this case, your acetyl group or your small molecules from stage 2 are further oxidized. And the end product of this oxidation is acetyl coenzyme and is now reduced to coenzyme NADH. And this stage involves numerous reactions which occur both in the cytosol as well as the mitochondria in the cell. Let's now proceed to stage 3 or your citric acid cycle. This one takes place inside your mitochondria and your acetyl group is oxidized to produce carbon dioxide and energy. And some energy produced in this stage is lost in the form of heat. And most energy is trapped in reduced coenzymes NADH and F and FADH2. And the carbon dioxide we exhale comes primarily from this stage. All right, the fourth one is electron transport chain and oxidative phosphorylation. This one also takes place in the mitochondria. Your NADH and your FADH2 are oxidized to release hydrogen ions and electrons. It is also needed for the production of your ATP, primary energy carrier, and in metabolic pathways. Your oxygen is also inhaled, is also converted into H2O in this stage. So, what are the stages of energy production in the all order of occurrence? Sige, kindly guess. All right, it's digestion, acetyl group formation, citric acid, electron transport, and oxidative phosphorylation. So your citric acid cycle, it is the series of biochemical reactions in which the acetyl portion of your acetyl coenzyme A is oxidized to carbon dioxide and the reduced coenzymes FADH2 and NADH rather, are produced. This is also known as your TCA or tricarboxylic acid cycle that has presence of 3 carboxylate groups in citric acid. It is also known for its Krebs cycle that is named after Hans Krebs, who elucidated this pathway. So there are important reactions in the citric acid cycle that includes reduction of your NAD plus your FAD to produce your NADH and FADH2. The carboxylation of citric acid will produce carbon dioxide. And yeah, this is now the summary of the citric acid cycle starting from your acetyl coenzyme A down to um, um, your last reaction of your GTP. So, um, this is now our citric acid uh, cycle. No? So this is now our starting point. So the cycle begins when your acetyl coenzyme A or your carbon 2 or your C2 combines with your oxaloacetate or your C4 to form your citrate or your C6. Through a series of 8 enzyme catalyzed reactions, your citrate is now oxidized, um, releasing carbon dioxide, generating NADH, FADH2, and adenosine triphosphate or your GTP, and generating oxaloacetate to restart the cycle. So you please take a screenshot. So let's now start with your condensation, in which there is your acetyl coenzyme A or your C2 plus your oxaloacetate, your C4, that results to your citrate or your C6. Now, what happens here is um, it is being catalyzed by citrate synthase, forming a 6-carbon compound. Okay. How about your isomerization, right? Or your isomers? Um, your isomerization is it is also coming from your citrate or your C6, moving to isocitrate. So what happens in this stage or in this cycle? Um, is now being catalyzed by aconitase. Again, it is now being catalyzed by aconitase, rearranges structure to prepare for oxidation. And for your third step, no, what happens in the oxidation and decarboxylation process? There is now again your isocitrate and then your alpha ketoglutarate or your C5 plus the presence of your carbon dioxide. Now, this one will now start to produce um, um, NADH. And then first is your carbon dioxide is being released. And for um, wait, and then for the oxidation and decarboxylation, we have an alpha ketoglutarate and succinyl coenzyme A. That's now yang you produce is NADH. The second one is CO2 is being released. Another one is the fifth one is phosphorylation. In this stage, it is now producing ATP. What else? Or JTP through the substrate level of phosphorylation. And on your sixth one, or your oxidation, it is catalyzed by succinate dehydrogenases, producing FADH2. And for your hydration, it is catalyzed by fumarase. It adds water across the wall band. And for the oxidation, it is catalyzed by malate dehydrogenase, producing NADH and regenerates the starting compound. So, uh, what is the biological importance of this citric acid cycle? This one is central to energy production and it actually provides most of the reducing power for ATP synthesis via electron transport chain. Also, it provides biosynthetic intermediates for amino acids, fatty acids, and other compounds. It also functions as the hub of metabolism, linking carbohydrates, fat, and protein at the catabolism. Sige. So let's now continue to the reactions of the citric acid cycle. So step one is formation of citrate, formation of isocitrate, oxidation of isocitrate, and formation of CO2. And step four is oxidation of alpha ketoglutarate and formation of CO2. I've mentioned it a while ago, but let me just show it to you. Sorry. And yeah, step five is thioester band, cleavage in succinyl coenzyme A and phosphorylation of GDP. Step six is oxidation of succinate. Step seven is hydration of fumarate. And step eight is oxidation of L-malate to regenerate your oxaloacetate. So, when one acetyl coenzyme A is processed through the citric acid cycle, how many times does each of the following events occur? Sige. All right. Your letter A is step six. Letter B is two. Step one and five. Letter C is four. Step three, four, six, and eight. Letter D is only one on the step three. Please, you can do a screenshot. All right. So let's have the regulation of your citric acid cycle. The rate at which the citric acid cycle operates is controlled by the body's need for the ATP. And when ATP supply is high, ATP inhibits cytase, or in the step one of the cycle. And when ATP levels are low, ADP activates citrate synthase. Similarly, ATP and NADH control isocitrate dehydrogenase, and your NADH acts as an inhibitor, and your ADP acts as your activator. So how many high energy molecules are formed in one turn of the citric acid cycle? All right, please guess your answer. Okay. How about your electron transport chain? This is a series of biochemical reactions in which intermediate carriers aid the transfer of electrons and hydrogen ions from NADH and FADH2. Ultimately, reacts with molecular oxygen to give H2O. Your electron transport chain also lost energy, is used to synthesize ATP in oxidative phosphorylation. Further, um, your enzyme and electron carriers for ATC located along the inner mitochondrial membrane. It's also organized into four distinct proteins complexes and two mobile carriers. Um, your protein complexes tightly bound to membrane. Your complex one is NADH. That is your coenzyme Q reductase. Your complex 2 is succinate. That is your coenzyme Q your reductase. Complex 3 is coenzyme Q or your cytochrome C reductase. And your complex 4 is cytochrome C oxidase. Your two mobile electron carriers is also known as the coenzyme Q and cytochrome C. So let's start with your complex 1 or your NADH coenzyme Q reductase. Your NADH from your citric acid cycle is the source of the electron process via this complex. It also contains approximately greater than 40 subunits, including flavine mononucleotide of your FMN and several iron, that is your sulfur protein clusters or your FeSP. And the net result is the transfer of electrons from your NADH to coenzyme Q. And there are several intermediate carriers that are involved in this electron transfer. And for your complex 2 or your succinate coenzyme Q reductase, it's actually smaller than complex 1 and it only contains 4 subunits including FeSPS. Succinate is converted to fumarate via this complex, and this complex is processes of FADH2. Coenzyme Q is the final recipient of the electrons from FADH2. And for your complex 3 or your coenzyme Q to cytochrome C reductase, it actually contains 11 different subunits and several FeSP proteins and cytochromes are electron carriers in this complex. Your cytochrome is a hemi-iron protein in which reversible oxidation of an iron atom occurs. And various cytochromes, such as your site A, site B, site C, and so on, differ from each other. Their protein substituents, the manner in which the hemi is bounded to the protein, and its attachment to the hemi ring. And we have the complex 4 or your cytochrome C oxidase. This is actually estimated that 95% of the oxygen used by cell serves as the final electron acceptor for your ATC. So let's have a practice exercise. So this is now your complex 1, 2, 3, and 4. And that's how the electrons flow. So question is, with which of the 4 complexes in the electron transport chain is each of the following events associated? Sige, please guess your answer. All right, A is complex 1, 2, 3, 4. Letter B is complex 2. Letter C is complex 4. And letter D is complex 3 and 4. How about this? Which statement best describes the electron transport chain? Sige, kindly guess. All right, that's the answer. So we are now down to oxidative phosphorylation, an introduction. Oxidative phosphorylation is a process by which ATP is synthesized from ADP using the energy released in the electron transport chain. And it can also be coupled with complex or other reactions. Coupled reactions is paired with biochemical reactions that occur concurrently in which energy released by one reaction is used in the other reactions. Examples is oxidative phosphorylation and the oxidative reactions of the electron transport chain. Coupling of ATP synthesis with the reaction of the ATC is related to the movement of protons and your hydrogen ions across the inner mitochondrial membrane. Your complexes 1, 3, 4 of ETC chain have a second function, meaning they serve as a proton pumps, transferring proton from the matrix side of the inner mitochondrial membrane to the intermembrane space. So for every electron pass through the ETC. Then, 4 protons cross the inner mitochondrial membrane through complex 1. And then, 4 through complex 3 and 2 more through complex 4. And this proton flow causes a buildup of hydrogen ion in the intermembrane space. And this high concentration of proton passing through ATP synthase becomes the basis for the ATP synthesis. All right. So let's have a practice question. What main event in oxidative phosphorylation is responsible for ATP production? What's your answer? All right. So how about your ATP formation? Your ATP formation for each mole of NADH oxidized in the ETC, we have 2.5 moles of ATP that are formed. And for each mole of FADH2 oxidized in the ETC, only 1.5 mole of ATP are formed. And for each mole of GTP hydrolyzed, 1 mole of ATP is formed. So if we will be um, getting the sum of all your ATP, it will now give us around 10 adenosine triphosphate. So how many moles of ATP are formed for each mole of NADH and FADH2? Okay, it's 2.5 and 1.5. Sige, balik na. 2.5 and 1.5. And how many moles of ATP are ultimately produced from the processing of 1 mole of acetyl coenzyme A molecules through the common metabolic pathway? It's approximately 10. Okay, so I think that ends our final lecture discussion. So, taman lang dari ato ang final examination question. So, if you have further questions, you can just message me, email me, so I can also respond to your different queries. So for now, kindly prepare for a quiz on site. So we can also assess your knowledge on this lecture. So I think that would be all, and have a great day, everyone. See you.